Welcome to this live broadcast of AP Bio Review with Mr. W. It is so great to have all of you here. Wow, it is so fantastic. What I would love for us to do as we get started is for those of you who are in the viewing audience to just say hello. I'm seeing a comment from Bryson that Bryson needs a 5. Where are people coming in from? Who are you? Where are you going to school? Stuff like that. Just tell me a little bit about yourself and also how you're feeling about the AP Bio exam. So let's fill that comment thread with just a couple of comments telling me who you are, how you're feeling about Thursday's test, and let me know a little bit about who is in the viewing audience. I am so grateful to you for spending your time with me on this Thursday. It's not Thursday. I'm sorry I said it's Thursday because probably Thursday has sort of a scary feel. What I meant is on this Monday afternoon, and I meant to go like, no. I meant to go like this. There I am. Yeah. So anybody just want to tell me who they are and where they're viewing from and how they're feeling about the upcoming test? That would be a wonderful way for us to get started. And why don't I just tell you what the plan is for the evening as we go ahead and do that. We're going to be together for a little bit under an hour. I've got another thing that's going on at 6:30. It's actually a private Zoom thing that you could participate in, but you'd have to sign up for learn-biology.com. And Morgan Lee from New Jersey. Okay. And it's so great to hear from all these people, Kavya and Glass and Coda, Cedar Park. I don't know what state that's in. North Carolina. This is fantastic. I am so grateful to all of you for joining me here tonight. It's so fun. And I'll tell you a little bit about myself in about 10 minutes, but you must have felt good enough about me to join this live review. And I'm also glad that I was able to do it a little bit earlier. I know that the time that I've been doing it, I'm in California, which is 7 p.m. It's probably a little bit late for some of you folks on the East Coast. So anyhow, I'm really glad to have you here. I put a asterisk next to the word evolution over here. And the reason why is because the College Board calls this unit natural selection. Bad name for the unit. It really should be called evolution because, as we'll see, a lot of evolutionary change isn't related to natural selection. That, of course, is incredibly important. It's kind of the foundational idea, but it's not the only idea. So I wish the College Board hadn't done that. Maybe write them an email after the test demanding change. So here's tonight's agenda. I'm going to move myself to this spot over here so that I'm not in the way. There we go. Okay. So tonight's agenda. First of all, I want to welcome you. You are now members of a community called Biomanians. And you can, after this show, you can go to mom, dad, whoever you're with, and just say, I'm a Biomanian. And who are Biomanians? Biomanians are people who care enough about biology to come to a live show on a Monday night to get ready for the exam. And Eliza from New York City. Fantastic. You know, for both the MCQs and the FRQs, I'll have some advice for you about both of those. But, you know, in general, let me tell you about what we're going to do tonight. Your questions, if you have like specific questions about specific aspects of biology, I could push my agenda to the side and answer any of those questions. But if we don't have that, then what we'll be doing is reviewing Unit 7. That's a good thing to do because Unit 7 is the core unit of the course. It's the longest unit of the course, and most of your questions will in some way impact evolution because evolution is the foundational idea of all of biology. About a week ago, I put out a 5-day plan. And of course, now you only have 3 days, and you kind of have less than 3 days depending on where you are. So you'd have to compress it. But I couldn't think of doing anything that was less than 5 days. But just go to apbiosuccess.com/5-day plan. Just, you know, you can type that in just as well as I was going to put it in the comment thread. But type that in. And here's really what the theory is, the theory behind your study process for the next couple of days. You want to find your gaps. Find your gaps. In other words, find the areas where you're not strong, where you don't know stuff. And I just want to say, like, if you're taking AP Bio, you're probably taking A Push and stuff like that. Like there's all these other courses. It's the same thing. The AP Bio, the plan is free. My website isn't free, but my website is laughingly cheap. It's $24.95 for a monthly subscription. And you're only gonna be with me for 1 month, right? You're gonna be with me for a week. In fact, you're gonna be with me for 3 days, and then you're gonna be done. So yeah, I mean, I don't give it away for free, but it's really reasonable, especially at this time of the year. So you sign up for the website for $24.95. You'd cancel when you were done on Thursday, and that would be your total expense. And anyhow, I'm going to tell you more about the website in a little bit, but right now, what I just want to say is, however you study, study actively. You want to be quizzing yourself. You want to be using things like flashcards. Flashcards are really the best. They're proven by cognitive science to be the best thing. So find your gaps, find the things you don't know. That's why I have a checklist, so you can go through. The checklist is connected to the plan. So get the plan, you'll also get access to the checklist. And then just keep on testing yourself, finding the gaps, repeat. Test yourself, find the gaps, repeat. Your study should all be incredibly active. And that actually means you should be going to the College Board's website and just doing FRQ after FRQ after FRQ. They're all there for free. Just go and Google AP Bio FRQs, and you'll see tons of them. And Ruby, that's if you're doing the right thing. And Ruby is saying that she's been practicing with the 2023 FRQs. That's exactly what you want to be doing. And the College Board is incredibly generous with this. They give you the FRQs, they give you the scoring guides, and they give you sample answers. And you should be looking at all of those. You know, think of it almost like you're preparing for a play. And the script that you're memorizing is your FRQ response. And just keep on doing that, and do that with as many FRQs as possible. And that's how you'll build the confidence that you need to. And, you know, you have time to work on 20 of these between now and the AP Bio exam. And again, I'm going to be doing one. I'm going to be doing 2 or 3 in the session, the Zoom session, but you got to sign up to do that. So unit reviews. No, Coda Coyote, they stopped after unit 6, but then I have this massive video, which is like the top 25 concepts. And in that, I have the most important concepts from unit 7 and unit 8. So ultimately, I've covered the whole course. I wish I was able to get to 7 and 8, but I just ran out of time. So I'm really sorry about that. But code it, find that, go to Science Music Videos, and you'll see that video. I think it's the most recent one that I released. And you know, there are chapter markers in the video, and you'll be able to get stuff about unit 7 and unit 8. So you got the whole thing, Coda. Okay, so let's get into it. All right. So topic 7.1 to 7.3, that's natural, artificial, and sexual selection. Natural selection is what happens in nature. Darwin, and you know, you don't need to know about anything historical in AP Bio exam. You could go into the test and not know who was Watson and Crick or Charles Darwin, and it would not affect your score at all. But Darwin happens to be the guy who figured this out. And he used artificial selection as his model for how natural selection happens. And in artificial selection, breeders select traits that they find desirable. And those are the animals that they allow to reproduce. He took that as the model for how natural selection happens in nature. And I'm going to walk you through that in a second. Sexual selection is a form of selection where there's selection for reproductively beneficial traits that make you more attractive to the other sex, for example. So like these, this is a picture that astoundingly I took in my neighborhood. You can see it's not out in nature. There's like a fence and like a dumpster behind there, but there are a bunch of turkeys. There are turkeys in Berkeley, California that were displaying, a bunch of males were displaying for this female. And this is a form of sexual selection. And basically the female chooses which male she gets to select. And the males basically therefore are selected for traits like tail feathers and wattles. Those are, I'll just say it, those are sexy features in turkeys. And so the turkeys that are most attractive to the female, those are the ones that get to pass their genes on. The females pass on their preference, the males pass on their traits. So you create this sort of positive feedback loop that leads to extraordinary things. Let's talk about natural selection. So here's a very simple idea. We have a visual predator. We have a population that has some variation. But over time, there's mutation, which emerges in any population, and there's mutation and selection. So it's easier for the birds to see the bugs that stand out, the beetles that stand out. Over time, there are less and less beetles that stand out, and the variants that have the best camouflage, those are the ones who get to pass their genes on until over time, the population is more adapted. So that's the basic idea. I'm going to spell it out the way that Darwin does. There's inherited variation from recombination and mutation. Actually, Darwin didn't know about those things. And then there's another thing, which is that more are born than ultimately get to survive. Even in slow populations, there are more offspring created that can survive to grow up. The survivors have a beneficial trait. Mutation creates new variants. Repeat that over and over again, and you wind up with extraordinary adaptations. Extraordinary as the human brain, as the wing of a bat, as the ears of an elephant. All of the incredible things, like for example, if we just go back to this slide, this is a leafy sea dragon. It's a kind of seahorse that lives in kelp, and it's evolved these appendages. Why? Because those that had these little fleshy appendages had better camouflage. An example that you probably learned about in your AP Bio class, we'll talk about in a minute, but there are 3 kinds of selection that occur that you should know about. The first is, and King, I'm not going backwards in AP Bio topics. I've been doing a whole series of reviews, and I've covered almost all the units. I've also released units about them. Because I didn't do videos about units 7 and 8, I thought that tonight and Wednesday, those are the last 2 sessions before the exam, I would do those units. So there we are. So again, not going backwards, I'm doing 7 because that seemed to be the gap in my contribution to your AP Bio review. So selection. Most organisms, populations, you have distribution around a normal curve. So, if this were height, short people, average people, really tall people. Well, selection that's directional pushes that curve in one direction. And I did a Unit 6 live stream, and I also have a really strong video about Unit 6. That was my last video. So you can watch the video about that, is my suggestion to you. Or even better, do the tutorial on learn-biology.com. But, again, that's a whole nother thing. Stabilizing selection is selection against 2 extremes. So like, for example, if you have animals that are camouflaged, what you wind up doing is you eliminate the ones that stand out and you push the population towards better camouflage. Stabilizing selection happens in traits like the weight of human babies. Small babies don't survive as well. Large babies don't survive as well, and sometimes their moms don't survive as well either. But babies that are average, they have the highest survival rate. So there's selection against both extremes. Disruptive selection is very hard to think of examples for, but what it would be involved would be a situation where the mean trait was somehow maladaptive, and then you'd wind up pushing the population to 2 extremes. By the way, folks, I just want to let you know the beauty of YouTube Live is that it's interactive. So if you have questions at any time, put them in the comment stream. That's why I'm doing this, so that I can respond. In most AP Bio classes, you probably did an activity about adaptive melanism with the rock pocket mouse. That's a single example of a much wider phenomenon called adaptive melanism. Melanin is the pigment that makes skin dark. Melanocytes are the cells that produce melanin. Adaptive melanin is the darkening of the body. It can be skin, fur, scales, feathers, wings, within a population in response to the darkening of the environment. And it occurs within a population as the gene pool evolves in response to some kind of natural selection. It's not something that happens in an individual's lifetime. Like, I don't know, to give a kind of silly example, I get nice and tan every summer. That's actually about melanin, and that's about melanocytes. But that's not what this is. This is about the population being selected for darker skin tone. It's usually in response to predation. Not always, but usually in response to predation. The selective pressure, usually predation. And what's cool about this is that if I were to show you a map of where these rock pocket mice live, what you'd see is that it's happened in many areas. It happens in almost any area where the mice have adapted to live on a dark, rocky background. What you find, though, is that the areas are often separated. And what happens is that the mice that lived there were originally descendants of lighter colored mice. But in the darker environment, there's natural selection for dark coloration. And in different areas, the variants are different. You can actually look at the DNA and you'll see that there are different mutations that lead to more expression of the melanin pigment. So again, at this point in the course, you're not only finding your gaps, but you're making connections. Like, how's natural selection selected to gene expression? That's something that I just did this second. That leads us to population genetics. And what I want to get out of the way right now is I want to demolish a big misconception. Population genetics is how allele frequencies change in gene pools. And the biggest misconception that students like you have is this: dominant alleles are not more common, nor over time do they become more common. The frequency of an allele is based on the advantage or harm that it confers to its associated phenotype or to random historical factors. It's not all about selection. And just as an example, this is Kyle Pasek. He's an actor, a very famous actor, and he has a condition called achondroplasia. Achondroplasia is caused by a dominant allele, but achondroplasiacs are relatively rare in the population. If you were to travel up to Scandinavia, go to Sweden, most of the folks up there have blue eyes and blonde hair. Those are both recessive traits, though both of those traits are complex, and it's not just one gene that codes for them. So again, that's a country where most people have the recessive allele. It's got nothing to do with dominance or recessiveness. It's got nothing to do with dominance or recessiveness has nothing to do with frequency of alleles. Any questions about that? Okay. Yes, that is true, King Mantra. It's called polydactyly, and that is also a dominant allele, and again, exceedingly rare. Hold up your hand. Almost everybody's got 5 of these. Having 6 is I believe a dominant allele, caused by a dominant allele. So population genetics is one of the more mathematical parts of the AP biology curriculum. You know, you need to know chi-square. That's in Unit 5. You need to know about the Simpson Diversity Index. That's Unit 8. And then recombination is also Unit 5. But here's the mathematical part of Unit 7. It's the Hardy-Weinberg equation. And there are 2 equations: P plus Q equals 1, P squared plus 2PQ plus Q squared equals 1. And what I recommend that students do, and I have a tutorial about this on learn-biology.com, is not to work directly with the equations, but to create what I call cross multiplication tables. So you can lay out P and Q like this. Just put them in a Punnett square: P, Q, P, Q. And that means this box over here is P times P, P squared. This one is PQ, 2PQ for the heterozygotes, and Q squared for the recessives. Many, many problems are covered in this way. Many problems involve you being told that like 49% of a population has a recessive trait. Once you hear that, you know that Q squared is 0.49. You put that in this box over here, and then you work backwards. The square root of 0.49 is 0.7. The square root of, then once you know 7, you know that, oh, P plus Q equals 1, 7, 0.7 plus what equals 1? Oh, 0.3. Well, then you put 0.3 over here. Then what's P squared? 0.3 times 0.3, 0.09. What's 2pq? It's going to be 0.21, 0.7 times 0.3 plus 0.21, 0.7 times 0.23. I think you can fairly expect there to be some math. You know, it's really hard. I taught the course for so many years. I retired from the classroom last year. I've seen chi-square. I've definitely, Hardy-Weinberg shows up. You definitely need to know to do what I just did, because it actually can show up in a multiple choice question in a pretty easy way. So that's how easy the math is over here. And again, it'll always be numbers that are pretty much easy to take the square root of. I mean, I can't guarantee that, but you know, so like 0.49, or maybe it'll be like 0.64, 64%. And then you know this is 0.8 and this is 0.2. Somebody asked a question about chi-square. I'm not going to cover that tonight. I covered that. Gee, I didn't really cover that in my Unit 5 tutorial. Here's what you would do. I'm just going to jump to my website and show you. So if you went to learn-biology.com, so it's really easy to navigate. You go to Courses, AP Bio, go to Unit 5, and then I've got a tutorial about how to do the chi-square test. And it's very short, and it's got practice problems and things like that. So, so Grayson, Golda, that is what I would suggest to you. And again, you can read all about it, and I explain most of it here, so you get most of it. But like, if you want to do these interactive pieces, that requires a subscription to the website. Again, it's $24.95. It's not a huge amount of money. I'm sure your parents will support you with that. So this is how you do it, and I sort of teach you how to do the whole thing within tables. So Grayson, I hope that is a reasonable response. I don't have a video about that. I'm sorry. So here we are. Okay, so that was this. I'm going to go there, click that. Beautiful. So in addition to the Hardy-Weinberg equations, there's a Hardy-Weinberg principle. All right. And King Mantra. Here I am going into it more in depth, just as you asked. So the idea is that the frequency of alleles and gene pools will stay constant unless one of the more following conditions is not met. So Hardy-Weinberg, they were two mathematical population geneticists, like they kind of invented population genetics, and they independently developed this fictional model population where evolution was not happening. Well, what has to be going on in that population? It's a non-evolving population. So it's not a real population, it's a fictional construct. It's infinitely large. There are no harmful or beneficial alleles. Everyone is mating randomly. There are no phenotypes that are more attractive than any others. No one's entering or leaving the population. It's a closed system. And there's no tendency of any one allele to mutate into another allele. So those are the 5 aspects of the Hardy-Weinberg equilibrium conditions in which a population does not evolve. And that is really cool. It's very powerful, because once you know that, then you can look at the factors that cause evolution. And those factors include genetic drift. That means that in populations that are very small, there can be random factors. Random sampling is what a statistician would call it, that would lead a population's gene frequencies to change over time. Natural selection, which we talked about at the top of the show, that can also lead to natural selection can lead to the change in allele frequencies. Like with those rock pocket mice, the allele that caused dark coloration increased in frequency over time. Sexual selection, we talked about that with those turkeys in my neighborhood, that because certain phenotypes were more attractive, then the allele that caused, for example, long feathers in those turkeys got selected for. Gene flow. when alleles from one population enter another population. And that can be by individuals moving in, or it can be by like pollen getting blown from one field to another, or directional mutation if one allele has a tendency to move to another. Sol FN. Holy cow, you're anticipating exactly where we're going. I'm now going to talk about genetic drift and how population bottlenecks work. Good job. So genetic drift is random change in allele frequencies in a population's gene pool, and it's usually associated with small size. So one form of that is a population bottleneck. And in the population bottleneck, imagine a sad scenario in which all of these gray pigs in this pig population died because of a viral infection. And the only pigs that were left were these two pink ones over here. Now, they didn't have an adaptation that enabled them to survive. It's random. It's random selection. And so the result is that this population now has lost many of its alleles. It went from being having a lot of, this is a big word, heterozygosity, to no heterozygosity. Everyone's a homozygote in this population. Some alleles might disappear completely. And again, the survivors didn't have any selective advantage. They were merely lucky. So that is called the bottleneck effect, population bottleneck. And this is called the founder effect. And that's another type of genetic drift. And that's when a small number of individuals from a large population found a new population. So, for example, this represents the mainland. And these are 3 colonies that are set up. And those could be human colonies, but those could also be like mainland and an island that gets colonized by birds or insects or something like that. Plants, for example. In this case, only red individuals, individuals with the red allele were the ones who made it over. So now red in this subpopulation is at 100%. The blue allele was lost. Why? Because all the founders only had the red allele. In this island over here, you maintain heterozygosity. There's the red allele and there's the blue allele. And in this island, the blue allele is lost. And a classic example of that is the Pennsylvania Amish, who have a high frequency of certain genetic diseases like Ellis von Creveld syndrome. Why is that? That's because they were founded by a relatively small number of people from the European mainland, and those people brought some uncharacteristic genes with them. So in the descendant population, which is relatively secluded from the outside population, there's a high frequency of that condition. Another phenomenon to note, I'm not going to go over all of the Hardy-Weinberg conditions, but sickle cell anemia is a really cool example of selection and evolution in general. In certain African populations, the allele for sickle cell anemia is extraordinarily high. Yet this is a disease that when you're homozygous recessive, it has really bad effects. They're not as bad as they used to be. And over the course of my teaching career, it went from like certain childhood death to now people living to their 50s and 60s all the time because of improvements in medical treatment. So sickle cell disease is caused by a point mutation in hemoglobin. That point mutation results in the amino acid valine, which is nonpolar, being substituted for glutamic acid, which is obviously acidic. That changes the structure of hemoglobin, that oxygen-carrying molecule, so that the cells sickle. And when they sickle, they get caught in capillaries and tissues, and that causes huge, huge pain crises and organ and tissue damage. You only have sickling in individuals who are recessive. Why isn't it removed for the population? Well, heterozygotes can experience a small amount of sickling, but not enough to cause the full-on effect. And here's the deal. In areas with the parasite malaria, malaria is a parasite that is so small that it can fit inside I called it malaria. Malaria is not the parasite. Malaria is caused by a parasite that can fit inside red blood cells. It's carried by mosquitoes. those red blood cells are hostile in heterozygotes for the Plasmodium parasite that causes malaria. So that leads to what's called heterozygote advantage. If you're big S, in other words, you don't have the sickling allele, you're selected against. Why? Because malaria can harm you. If you are big S, little s, you have the best phenotype. Because you have this phenotype that protects you against malaria. And if you're little s, you are selected against. Okay, so there's selection for heterozygotes, and there's a high frequency of that allele. Model might apply to some other diseases like cystic fibrosis and Tay-Sachs. How does this relate to alpha wolves? And I don't know how this relates to alpha wolves. Puzo, I think that's a different thing. I think that mostly relates to things like social dominance hierarchies and stuff like that. So it's a little bit out of the scope of this. It might actually fit more in Unit 8's first topic, which is about animal behavior. Sorry, I don't have a better answer, Puzu. Okay, just want to take a minute to introduce myself, since I think many of you are new to me and these shows that I've been doing. My name is Glenn Wilkenfeld. I'm a retired AP biology teacher, but really recently retired. I only retired last June so that I could do this full time. And then I create videos on my Science Music Videos channel, which I've been doing for a super long time. Some of my videos go back to way before you were born. I'm also known as Mr. W. I taught AP biology for a long time, and I was super successful in helping kids get 4s and 5s. And Ruby, one of the FRQs I did was talking about hemoglobin and myoglobin binding to oxygen. It had something to do with how muscles use oxygen. Hemoglobin relates to oxygen normally. Ruby, the basic idea is that hemoglobin is the molecule that carries oxygen in red blood cells. I don't think you need to know much more than that. A cool thing is that hemoglobin is a quaternary protein. It's got 4 globin units that are connected together. But myoglobin is a molecule that's in muscle tissues that's probably evolutionarily related to hemoglobin. And myoglobin can hold a little bit of oxygen in muscle tissues when they're doing aerobic respiration. Ruby, I don't know if you need to know more than that. If you gave me if you knew the year, I could possibly look that up and then give you a better answer for the next show. And Morgan, thank you very much for that comment. That is really appreciated. Thank you very much. So I was just introducing myself and telling you that I had a lot of success teaching AP Bio. And one thing that I did was I created a website that is an interactive AP Biology curriculum. And I think this would still be useful to you. I know it's only 3 days before the AP Bio exam, but you can do a lot of great studying on learn-biology.com. Why? Because we've got over 450 AP Bio review flashcards. So again, remember I was talking about like find your gaps, fill them. Use the flashcards to fill the gaps. Find your gaps. You can simultaneously find your gaps and fill them with our FRQs because they have questions and then answers. And you look up the answers and you would see how you were doing on those FRQs. And then we've got multiple choice questions, which are fabulous because they're interactive. So if you got a question wrong, you get a hint about the answer and it'll really build your understanding. And then we've got these super fun games that are called click-on challenges. And not only that, but for the same $24.95, you also get access to the Biomania AP Bio app. So, you know, there are many of you listening right now. You would all benefit by using learn-biology.com, though, of course, many of you might already be using it because there are hundreds of teachers around the country who use it as their textbook and all their students are using it. So we've got 8,500 students who are using it right now. And really my message is to students whose teachers aren't using it and you could use it. Yeah, you got to know how to do recombination frequency. And I have a tutorial about that on learn-biology.com. You know, there's 2 water potential formulas. There's an easy one, which is, you know, psi equals psi s plus psi p. You might have to deal with that one. The other one, which is how you figure out just solute potential, it's on the formula sheet. And listen, you'll be mad at me if it's not, if like I say it's not important, but I can't imagine that it would be on the test. It just would take a lot of time. So like know the easy water potential formula, not the hard one. And then know this thing. If you're told that you're looking at a data set, I'm going to make myself a little bit bigger so that you can see this a little bit more easily. I'm going to go like this. Okay? So error bars. Here's what you need to know. If the error bars, I'm going to try and position myself good. This is 1 point error bar, another point error bar. This is a statistically significant difference. If there's overlap to any degree, then it's not statistically significant. And I was going over an FRQ with a bunch of students last night, and they actually had this. And there was a data set where like 2 of the data points were like this, not a statistically significant difference. I don't think you'll need to calculate statistical, like the statistical difference or anything like that, but definitely chi-square, no chi-square, and know how to do recombination. I got a tutorial about recombination. That's pretty tough. The N terminus and the C terminus, I kind of forget about that. I got to tell you the truth. One N terminus, that's like one end of a polypeptide, and then the C terminus is the other end of the polypeptide. I don't think you have to worry about it. And I'm sorry that, like, I could stop and like look it up for you right now, but I wouldn't worry about that as much as I would big picture stuff. Storing equations on the calculator because college. Morgan, I don't know that, but I'm sure that stuff is publicly available and you can look it up. And again, I'm 1 year out of the game, so I'm not, I don't have students asking me about that right now, but you can find on the College Board's website about like what kind of calculator you can bring, and just don't take any chances with it. I used to give my students, I sort of had a class set of 4 function calculators, and for kids who didn't have one, I would give them that. That's it. Though I know you can use a scientific calculator. All right, so folks, I just want to tell you a little bit about myself and what I had to offer, but now we can go back to evolution, and we've only got about 20 minutes left. So let me go like this, and now I'm going to go all the way like this. Much better. Okay. Evidence for evolution. The most important thing to know about with evidence for evolution, like you need to know that like there are fossils, but you need to know a lot about homologous structures. That's a really big evolutionary theme. And homologous structures are ones that are derived from a common ancestor and then get modified in different ways through a process that's called adaptive radiation. Adaptive radiation is when an ancestor, like imagine this ancestor over here, one line diverging to humans, one line diverging to dogs, one to birds, one to whales, all right? And these are in kind of a funny order because the birds would be the least related to the other ones. But in any case, that's homology. The fact that like these bones are the same bones. And not only can you tell that anatomically, but they also develop from the same tissue in the embryo. And that's another clue that there was once a common ancestor, and then that ancestor diverged in different ways. Very, very important evidence. Another kind of evidence for evolution is vestigial structures. Those are structures that over evolutionary time have lost their function. Homologies can also be on a molecular level. So like, for example, oh, this is so great. Here's hemoglobin. I know it's really, really small, and I'm sorry about that. But what you can tell is that the hemoglobin of a human and the hemoglobin of a gorilla are almost exactly the same. Out of 146 amino acids, there's only one difference. Only one difference. So that speaks to the evolutionary closeness between humans and gorillas. And then you can actually make predictions. Like you would predict that of these animals, the most closely related one would have the most similarity in terms of amino acid structure. And it turns out that the monkey, also a primate like us, has only 8 differences. And as the animals become more evolutionarily distant, their the number of amino acid differences increases. And then finally, there's natural selection that we've observed over time. And the biggest example of that is resistance. So like, for example, mosquitoes have become resistant to pesticides like DDT. Bacteria have become resistant to antibiotics. Why? Because at the start, there's variation. The pesticide kills most of the mosquitoes, except for the ones that have slightly more resistance. And they're the ones who stagger back and they wind up reproducing and they pass on whatever genes they had that made them slightly more resistant. So over time, you have the increase of resistance. In bacterial populations, which reproduce every 30 minutes, you can have the growth of resistance happening incredibly quickly. And that's why we're losing the use of our antibiotics over time. So these are all pieces of evidence for evolution. And these, I think, are the main ones to be familiar with. Fossils, homologous features, vestigial features, molecular homologies, and continuing evolution. You need to know how to interpret a phylogenetic tree. I actually think that's one of the most fun aspects of biology. But like, for example, you need to know key concepts like a clade. A clade is a group that consists of all of the organisms, all of the species that are derived from a common ancestor. So like, for example, group 6, that's a clade. And that clade consists of the woodpecker finch and the small tree finch. Here's another clade, the common cactus finch and the large ground finch. And here's another clade. All 4 of these are derived from a common ancestor. Ruby, thank you very much for that question. A homologous structure is one that's derived from a common ancestor. So like, for example, all of these finches are related. Their beaks are homologous. They've diverged over time due to adaptation to specific food sources. That's a homology. An analogy is a structure that is superficially similar, but it has evolved from different evolutionary origins. So like, for example, sharks and dolphins, they have a very similar shape. That shape is analogous. Sharks over here, dolphins over here, they evolved this similar shape. Why? Because they're both adapted to swimming fast in the water, right? But they didn't inherit that from a common ancestor. So that's the difference right there. And Yera, I don't think there'll be a lot of Hardy-Weinberg calculations, but I think that it regularly shows up on multiple choice questions and sometimes on FRQs as well. And what unit do you think will, and Yera, it's, you got to know the whole course. Unit 7 is the biggest unit in the course, but you got to know the whole thing. All right, let's go on. Clades are the most important concept. A node is like the branch point between 2 things. A shared derived feature. So whatever feature it is that binds this clade together, that's a shared derived feature. I'm trying to think of an example that will mean something to you. So like, for example, a shared derived feature of all the primates, so that means like us and the monkeys and the apes, is a rotatable shoulder with a ball and socket joint. I mean, think about like what would happen to your dog if you tried to rotate the dog. Dogs can't do this, right? That's because they're outside of our clade, the primate clade. They're in another clade. They're the carnivore clade. So that's a shared derived feature, okay? Very, very important concept. An ancestral feature is a feature that's shared by all these organisms, but it's also shared by larger clades that they're a part of. So like, for example, we're mammals, right? A shared derived feature of the mammals is producing milk and having hair on our skin. Those are both shared derived features. What's an ancestral feature? Having a bony skeleton with a backbone. All mammals have that, but so do reptiles, so do fish. So it's a feature that we share, but it's also shared by other groups in a larger clade that goes wider than just the mammals. So that's the deal there. And this idea of molecular clocks, like I was just working on an FRQ with a bunch of students where you had to calculate time of divergence. Leo, of course you're not taking the test. Folks, I have a former student of mine who just joined this, and that makes me so happy just to see Leo again, my student from last year. So great. Okay, so I think we have just enough time to like cover one or two more concepts. Leo, you think all these folks should use learn-biology.com? If they're not in a class, it's going to cost them a little bit. So I just want to know if Leo, if you're still listening, maybe you can give learn-biology.com a little comment shout out. I know that's a lot of pressure on you. I'm just going to keep talking. Oh, there we go. Okay. So folks, just so that you know, you've heard it from a student. And again, I don't give it away for free. It's $24.95, but that's not a lot. And hopefully you'll use that, because if you got to cram for the next 3 days, this is the way to do it. And so let's do this. Ruby, that's a great question. I'm going to go back a frame. This is going to be a little bit hard to do, but if this had a scale over here, and they told you that the woodpecker finch and the small tree finch, that they're, based on fossil evidence, their time of divergence was 1 million years ago. All right. Then what they would do, then you'd sort of like look at that distance, and then you say, oh, double that, that would be 2 million years ago. But that's not how the question would go. You'd be told that the woodpecker finch and the small tree finch, that for a specific protein, their amino acid sequence differs by 2, and that based on fossil evidence, they diverged 1 million years ago. So that means that every single amino acid difference happens in 500,000 years, right? So there are 2 differences that happened 1 million years ago, right? So 2 differences would take 1 million years. 1 difference would take 500,000 years ago. And then they'd give you another animal that had 6 differences. And if every difference takes 500,000 years, then 6 times 500,000 years, then it would be 3 million years. So that's the deal. And Emma M., that's such a nice thing for you to say. And that's so fabulous. And I'm so happy to have a college student who's watching this. So Ruby, I hope that was a reasonable answer. And again, I know that it sounds like I'm selling my website, but I kind of am. You can find a tutorial about that on learn-biology.com, and that's how you would like solidly know how molecular clocks work. Where were we going? We're going this direction. So for speciation, you need to know the biological species concept. You need to know about reproductive isolating mechanisms, prezygotic barriers, and postzygotic barriers. The biological species concept is that like a species is a population that can interbreed with one another successfully, but can't breed with other groups. And this is so fabulous. I've got a lot of former students who are here. That's making me really happy. We've got reproductive isolating mechanisms. Those are things that keep the closely related species from interbreeding. And why not? Yeah, listen, the College Board practice exams are a good representation, but as far as I know, there's only one that's been released. That's a 2013 exam. It's 10 years old. And then at the end of the College Board course and exam description, you can get some more questions, about 25 more questions. And again, the problem is that you can't really get that many questions. So that is why, that's why people like me, we write our own questions. We try and simulate the difficulty of the College Board questions. But, the best thing to do for you to be doing right now is to be working with authentic College Board materials. But the next best thing is to use my website. The FRQs, Bryson, are totally right. The FRQs are fabulous, and the College Board is totally generous about the information that they give you. So we've got prezygotic barriers, we've got postzygotic barriers, and allopatric versus sympatric speciation. Allopatric speciation is when you have a population and it gets cut into two by a geographic barrier. On each side of the barrier, there's going to be all the things that we talked about, natural selection, and then all those population genetic things like genetic drift. That's going to lead to genetic differentiation over time. These circles represent alleles that have become very different. Over time, these two populations will evolve so many differences that when the barrier goes away, there will no longer be the ability of those two subpopulations to interbreed. So that's what allopatric is. Sympatric speciation is when speciation occurs without a genetic barrier. That is readily explicable in plants. Plants can do things where, like, for example, an error in meiosis will lead in one generation a species to double its chromosome number. And once that happens, the daughter species can't interbreed with the parent species anymore. So that's what we've got for allopatric versus sympatric speciation. This kind of graph over here is really important to understand as an important pattern in evolution. Every once in a while, due to things that are not connected to either population genetics or natural selection, but really more connected to astronomy and geology, the planet undergoes a mass convulsion and huge numbers of species are wiped out. Those are called mass extinctions. They lead to massive decreases in biodiversity. Following that decrease in biodiversity, the lineages that survive undergo an adaptive radiation and spread out. Up to 65 million years ago, the dinosaurs reigned on Earth. When that asteroid smacked into the Gulf of Mexico, it basically caused a mass extinction. Most of the dinosaurs, excepting birds, were wiped out. Birds are dinosaurs biologically. But then, like this line, that might represent the mammals over here. And then we underwent an incredible adaptive radiation into all the various forms of mammals that we have today. Yeah, viable means able to survive. So in the biological species concept, you have to be able to produce viable and fertile offspring. If you can grow up, you're viable. If you can grow up and you can reproduce, you're fertile. The example that sort of proves the rule is that if a horse and a donkey interbreed, they produce a mule. A mule is viable, but it's not fertile. Viable, but not fertile. And that means that there's a reproductive barrier between horses and donkeys. And that's actually called a post-zygotic barrier because obviously a zygote had to form. And Ivan, I really appreciate that. And folks, I'm really sorry, but we're out of time. And I told you I have this, I do these private Zoom meetings with students who have a subscription to LearnDash-Biology. And I'm just going to jump a couple of slides forward. Sorry, we have to skip Origin of Life and this, but I just really want to encourage you. If you're going to cram, invest in the best kind of cramming that you can do. It'll cost you $24.95, one-time cost. It's a monthly subscription, but you can cancel it anytime. Use my plan. Use LearnDash-Biology. And I think that's the best way that you can spend your time for the next 3 days getting ready for the AP Bio exam. And I really appreciate, Pranya, what you're saying because biology is the best. I spent my entire adult life teaching it and I retired and I'm still teaching it. So sign up to boost your score. That's where you're going to get the flashcards, the FRQs, the multiple choice questions, the click on challenges, the Biomania AP Bio app that you need for success. So folks, it's been wonderful spending this time with you. I hope I see some of you on learn-biology.com. Please sign up right now. Take out your phone, take a picture of this page, show it to your folks and say, Mom, Dad, $24.95, please. All right. Okay. And folks, it's been great. Everybody, be well. It's been great to all have you here. And I'm going to do another live stream on Wednesday night, I think at about the same time. So everybody, be well. And I actually just saw one payment come in. So maybe we can get a couple of more. I hope that was someone who was watching the show. Again, it's the best investment you can make in the next couple of days to really boost your scores in biology. Folks, I got to go. Everybody be well. Ivan, I'm having the greatest time. I get to do this all the time, but I do miss students. So that's why I'm here. Folks, everybody be well. See ya.